In this question, you have to identify the equivalent of a structural gene. Option A, operon. It is incorrect because an operon refers to a cluster of genes under the control of a single promoter and regulatory elements functioning together in prokaryotes, and it includes structural genes, but it is not equivalent to an individual structural gene. Option B, recon. It is also incorrect because it represents the smallest unit of recombination in a gene, but it is not equivalent to a structural gene. Option C, muton. It is also incorrect because a muton is the smallest unit of mutation within a gene, not equivalent to a structural gene. Option D, cistron. A cistron is a segment of DNA that codes for a single polypeptide or RNA molecule. So, this is the correct option because this is equivalent to a structural gene. In this question, you have to identify the wrong statement regarding a molecule that can act as a genetic material. Option A, it should be unstable structurally and chemically. Now, this is not true because genetic material must be stable both structurally and chemically to ensure accurate transmission of genetic information across generations. Hence, it is the correct option. Option B, it should provide the scope for slow changes that are required for evolution. Now, this is true. Genetic material should allow mutations, which are slow and heritable changes, providing the basis for evolution. Hence, it is incorrect option. Option C, it should be able to express itself in the form of Mendelian characters. This is also true because genetic material must be capable of encoding information that is expressed as phenotypic traits following Mendelian inheritance pattern. Hence, it is incorrect option. Option D, it should be able to generate its replica. Now, this is also true because replication is essential for genetic material to pass on information to offspring during reproduction. Hence, it is incorrect option. DNA-dependent RNA polymerase catalyzes transcription on one strand of the DNA, which is called option A, alpha strand. It is incorrect because there is no term such as alpha strand in context of transcription. Option B, anti-strand. It is also incorrect because there is also no recognized term as anti-strand in the context of DNA strand or transcription. Option C, template strand. Now, DNA-dependent RNA polymerase uses the template strand of DNA to synthesize RNA. And this strand is complementary to the RNA transcript. Hence, it is the correct option. Option D, coding strand. It is incorrect because the coding strand has the same sequence as the RNA except for thymine, which is replaced by uracil, but it is not directly used during transcription. In this question, you have to identify the correct statement regarding thalassemia and sickle cell anemia. Option A, both are due to quantitative defect in globin chain synthesis. Now, thalassemia involves a quantitative defect, that is, either reduced or absent production of globin chains. But sickle cell anemia is caused by a qualitative defect, that is, a structural mutation in the beta globin chain. Hence, it is incorrect. Option B, thalassemia is due to less synthesis of globin molecules. Now, thalassemia results from reduced or absent synthesis of either alpha or beta globin chains of hemoglobin, leading to imbalanced or less synthesis of this globin production. Hence, it is the correct option. Option C. Sickle cell anemia is due to a quantitative problem of globin molecules. Now, it is also incorrect because, as already stated, sickle cell anemia is not caused by quantitative defect. It is due to a point mutation in the beta globin gene, causing a structural change in the hemoglobin molecule, making it to be appear like sickle-like. Hence, it is incorrect. Option D. Both are due to a qualitative defect in globin chain synthesis. It is also incorrect because sickle cell anemia is due to qualitative defect, but thalassemia involves a quantitative defect in globin chain synthesis. In this question, you have to identify the experiment that paved the final proof for DNA as the genetic material. Option A, Hershey and Chase. The final proof that DNA is the genetic material came from the Hershey-Chase experiment in 1952. They used bacteriophages, that is, viruses that infect bacteria, and labeled it with radioactive isotopes of phosphorus and sulfur, P32 for DNA and S35 for protein, to demonstrate that it was the DNA of the virus and not the protein that was injected into the bacterial cell and carried the genetic information. So, this experiment provided a strong evidence that DNA is the genetic material. Hence, it is the correct option. Option B, Avery, MacLeod and McCarthy. Now, in 1944, Avery, MacLeod and McCarthy showed that DNA was the transforming principle in bacteria. That is, it carried the genetic information. But their work was not the final proof, though it was a significant contribution. Hence, it is incorrect. Option C, Hargobind Khorana. It is incorrect because he is known for his work in understanding or regarding the genetic code and its translation into proteins. Option D, Griffith. Now, Frederick Griffith performed an experiment in 1928 that suggested the existence of a transforming principle in bacteria Streptococcus pneumoniae, but he did not identify what was the transforming principle. Hence, it is incorrect. In this question, it is given that there are 999 bases in an RNA that codes for a protein with 333 amino acids, and the base at position 901 is deleted such that the length of the RNA becomes 998 bases. So you have to identify how many codons will be altered. Now it is given that the RNA has 999 bases and it codes for 333 amino acids. Now there is deletion of one base at position 901 and the RNA sequence becomes 998 bases long. And this deletion causes a frame shift mutation, that is, altering the reading frame from the point of deletion onwards. Now, so the effect of the frame shift mutation would be that the codons before position 901, that is 900 divided by 3 equal to 300 codons, remain unchanged. The codon at position 901 and all its subsequent codons would be affected. So from position 901 onwards, there are 999 minus 900 divided by 3 is equal to 33 codons. So the number of altered codons would be 33 codons. Since the deletion shifts the reading frame, so all these 33 codons from position 901 onward would be altered. So the correct answer is option B. During replication, the Okazaki fragments are used to elongate in which direction? Now, during replication, the leading strand is synthesized continuously in the direction of the replication fork, and the lagging strand is synthesized discontinuously in short fragments which are known as Okazaki fragments, because DNA polymerase can only synthesize in 5' to 3' direction, and these fragments are synthesized away from the replication fork, and later they are joined by the enzyme DNA ligase to form a continuous strand. Hence, the correct answer is that the lagging strand away from the replication fork. Hence, the correct answer is option C. In this question, you have to identify which type of RNA is the most abundant in animal cells. Option A, tRNA. It is incorrect because transfer RNA, that is tRNA, is the second most abundant. It comprises of about 10 to 15% of total RNA. Hence, it is incorrect. Option B, mRNA. It is also incorrect because messenger RNA, that is mRNA, is less abundant. It comprises nearly 3 to 5% of the total RNA. Option C, miRNA. MicroRNA, that is miRNA, it regulates gene expression during the post-transcriptional level, and it is present in very low amount compared to other RNAs. Hence, it is incorrect. Option D, rRNA. Now, ribosomal RNA, that is rRNA, is the most abundant RNA in cells, including animal cells, and it comprises of approximately 80 to 85% of the total RNA. And it is the structural and functional component of ribosomes, which are very essential for protein synthesis. Thus, it is the correct option. In this question, you have to choose the option that correctly represents what does the association of histone H1 with a nucleosome indicates. Option A, DNA replication is occurring. It is incorrect because histone H1 is not involved in DNA replication. Option B, the DNA is condensed into chromatin fiber. Now, histone H1 binds to the linker DNA between nucleosomes, and it helps in the compaction of the chromatin into a 30 nanometer chromatin fiber, thus facilitating the DNA condensation. Hence, it is the correct option. Option C, the DNA double helix is exposed. It is also incorrect because the presence of histone H1 indicates that the DNA is compacted or condensed and it is not exposed. Option D, transcription is occurring. It is also incorrect because transcription requires chromatin to be less condensed to allow the access of RNA polymerase as well as transcription factors. So the presence of histone H1 indicates a more condensed state, which is transcriptionally rather inactive.